So when we think of biochemical pathways, we think of reactions that begin with a certain chemical and then they end with a chemical product. So the energy sources for organisms in which these biochemical pathways works are going to stem from nutrients that are taken up by the organism. And then once those bonds that are made up of the chemical composition of these nutrients are broken, then energy can be released as well as stored inside of the system. The primary molecule that is great for doing this is that of glucose. And glucose can be catabolized by the processes of aerobic respiration, which is also known as cell respiration, or the process of fermentation, which is the conversion of a sugar molecule into essentially ATP, but it releases alcohol and gas products through this process. Both of these processes, both cell respiration and fermentation, start with a precursory process which is called glycolysis. And glycolysis is the simple splitting of a glucose molecule. This little diagram will show you, let me try to bring it up where you can see it, kind of a model of what that would look like. So you have a glucose molecule. Remember, glucose, when we talked about our molecular structure, consists of 6 carbons joined together. So you're 1, 2, 3, 4, 5, 6. Consider each of those circles a carbon. And then the cell will utilize an energy rod. We'll refer to that as ATP. And ATP will basically be split apart, where we release one phosphate in order to break apart this glucose molecule. Once the glucose molecule is broken apart, you end up having 2 chains of pyruvic acid, because that's what happens when it splits. Now what you're being shown in the diagram is a step-by-step process of how that happens. So you have the molecule adenosine triphosphate. A phosphate is released, and that phosphate group is affixed to your glucose molecule. So then the molecule is no longer referred to as glucose. It's referred to as glucose 6-phosphate. What this means is you have a phosphate group located on the 6th carbon of this glucose molecule. Okay, and as this process occurs, the ATP is converted from adenosine triphosphate to adenosine diphosphate. Once we have this phosphate glucose molecule, another ATP is brought into the system, and it's going to phosphorylate the other side of the glucose molecule. Okay, so now we have a phosphate group situated on our carbon 6, and we also have a phosphate molecule situated on our carbon 1. This molecular structure is now referred to as fructose 1,6-biphosphate. Okay, so that's the new name of the model, of the molecular model. I'm going to try to get rid of this text so that way we can look at this image a little bit more distinctly. Okay, so now that we have basically a phosphorylated glucose molecule, even though these have different molecular names to it, it really is just glucose that's containing 2 ATPs, or 2 phosphate groups, sorry, from 2 ATP molecules. Now we're able to split this molecule into 2 forms of pyruvic acid, and those are called pGal. So once the bonds between these 2 carbons are broken, energy is released, and you get these 2 molecules here known as pGal, where you have 1 phosphate group on the 6th carbon, or the original 6th carbon, and another phosphate group on the 1st carbon. But now you only have 3 carbon atoms inside this new pGal molecule. So the next step in this pathway is to enable this particular molecule to be ready to be converted into pyruvic acid. And so there's a series of biochemical steps that take place here. So one such step that occurs, well actually the steps that occur just occur in duplicates. Okay, so this pGal is going to undergo the same process as this pCal, so everything is essentially doubled. So in the process of converting this p-Gal molecule into pyruvic acid, or 3 sugar molecules of pyruvate, ADP, which was generated at the conversion of ATP, is also going to phosphorylate the p-Gal molecule. Before that happens, though, we have a phosphate group that is added to, so we have a hydrogen molecule actually that is going to be lost from the p-Gal that gets picked up by an electron carrier molecule that's known as NAD+. And the NAD+ is going to pick up or attract that hydrogen. So a hydrogen is lost here on this carbon molecule, because remember, carbon is bound to hydrogen, so that way that bond is shared. But with the loss of that bond, that enables a phosphate group to bind to this 3-carbon linear structure. So the phosphate group ends up getting added, and then adenosine diphosphate, which was generated here in this step, is going to pick up a phosphate group from the molecule. And when it picks it up, ATP is going to be generated. Okay? So another adenosine diphosphate molecule is to come by and pick up the other phosphate group, and again we have ATP being generated. So when the phosphate groups are removed, it converts the molecule now from this p-Gal molecule into pyruvate. And this happens twice in the cycle in the process that we know as glycolysis, which is the simple breaking apart of this glucose molecule. However, this is only made possible when you have the phosphorylation of the molecule, which means you're adding phosphates coming from ATP, and when you have the loss of this hydrogen from this electron carrier molecule known as NADH. Okay, so biochemical pathways are going to exist in a series of steps, just like I showed you here in this example of glycolysis. And these steps are only made possible, again, when you have the existence of certain enzymes to catalyze these reactions. In this image here, this is just showing you or outlining how one compound, which is compound A, gets converted all the way into compound E, which again is basically what I showed you in this particular diagram, where we have what you could consider to be compound A being which would be glucose, being converted into compound E, which is the pyruvate. So this happens when you have enzymes that react on these pathways and enable the conversion to continually take place. So enzyme 1 will operate on component A, enzyme 2 operates on B, enzyme 3 operates on component C, enzyme 4 operates on component D. And then as these structures continually change, you're able to be converted from compound A into compound E. So this is essentially the basis of what a biochemical pathway is going to look like. Now, in the process of catabolism, especially that of glucose, through the process of aerobic or cell respiration, we have essentially 3 pathways. We have glycolysis, we have the Krebs cycle, and we have the electron transport chain. I pretty much walked through glycolysis with you already when I showed you this diagram of converting glucose into pyruvate. So all glycolysis is the splitting of a glucose molecule and the conversion of it from component A, which is glucose, into component E, which would be your pyruvate. The next step of this process is to take this pyruvate and enter it into another series of biochemical pathways, which is known as the Krebs cycle. And in this cycle, essentially, you have, once this takes place, the end products of the Krebs cycle consist of NADH and FADH2, as well as hydrogen ion elements. And these particular items are very useful in the next phase of the catabolic pathway of a cell, which is the electron transport chain. And so in the electron transport chain, we essentially have carrier molecules moving electrons down a chain in order to generate ATP. So let's kind of delve a little bit into some more details of these processes. Again, the first phase is that of glycolysis. This particular process is considered to be anaerobic, which means oxygen is not required for the breakdown of a glucose molecule. Now, this differs from the Krebs cycle and the electron transport chain because both of those require oxygen. And so the entire process is going to be considered anaerobic, it's going to be considered aerobic because the Krebs cycle and the electron transport chain are, there's 2 out of the 3 that are going to require oxygen being present for cell respiration to take place. And when I say cell respiration, it's the same when I'm talking about aerobic respiration. I just prefer to say cell respiration. So glycolysis we walked through when I showed you this diagram here. This is a 9-step biochemical pathway, and none of these particular processes in breaking down the glucose molecule can occur without the presence of a specific enzyme being involved. And again, here is another diagram kind of outlining the entire process of cell respiration. There's a lot of nitty-gritty details that are involved in the process, but I don't expect you to know things like oxaloacetic acetate or citrate or isocitrates or any of those intermediate forms that take place in, say, like the Krebs cycle. But I do want you to understand and be familiar with the 3 primary processes and in general what happens. So in the process of glycolysis, we have the breakdown of glucose. So glucose is going to enter the cytoplasm of a cell as it passes through the cell membrane. It will undergo the process of glycolysis where it is converted into 2 molecules of pyruvate, and in doing so 2 ATP molecules are generated in that process. And again, if you spend some time studying this diagram, that'll help you to remember that you have 2 ATP products that are generated in the process. It's 2 ATP products that are generated taking place here. Overall, it's actually 4, but remember when you start this process, you end up using 2 of them. So really your net yield from the process of glycolysis is going to be 2, since it cost you 2 ATP in order to generate 4 ATP. So once we have the molecules of pyruvate, now we can go, a cell can undergo 2 pathways. And since we're undergoing the processes of aerobic respiration, we're going to go down the pathway in which oxygen is present. Okay? If oxygen is not required, then the pathway of fermentation is the selected pathway to generate energy for the cell. But when oxygen is present, then we enter the Krebs cycle, where we're going to have the byproduct of carbon dioxide being released. So this is one of the reasons why we breathe or exhale carbon dioxide, because it's a waste product generated from our Krebs cycle. And then from the Krebs cycle, we're going to enter the electron transport chain. And all of this occurs inside the mitochondria organelle of a cell. Okay, so with the electron transport chain, we also have to have the presence of oxygen in order for this to work, because you essentially have electrons being passed from carrier molecule to carrier molecule, and those carrier molecules were generated in the Krebs cycle. So as electrons are being passed, they're reacting with the oxygen that's present, and that's going to release water as well as generate 34 to 36 molecules of adenosine triphosphate, which is a lot of fuel molecules that the cell can use to continue this process, as well as to build structures and destroy and degrade large complex molecules inside a cell. So again, here is glycolysis, that same scheme that I've showed you. You start off basically by using phosphates from adenosine triphosphate, adding them to the glucose molecule, and then through a series of these biochemical reactions, you end up generating 2 molecules of ATP for each pyruvate that undergoes this process. And so this means that you use 2 molecules of ATP when you are constructing fructose 1,6-biphosphate And so you have a net worth of 2 ATP being generated in this process because 2 are used and